So our next speaker is Hendrik Dietz from Technical University in Munich. I suppose it's interesting to note that after the revolution comes the exploitation. To me, that's quite analogous to earlier industrial revolutions. So we're going to have some mention of nano cages, I believe, as I've looked at some of Hendrik's work, and he likes to put viruses in prison.
Thank you very much.
So the alarm will go at 11, so you'll know it's time to stop.
Got it. It's not working, right?
No?
Oh, now it works. It's just a matter of force. Okay, so hello everybody. Pleasure to be here. I guess I won't have time really to talk about motors and things like that, so I'll focus mostly about putting viruses in quarantine and things like that. So I want to spend a few seconds just sharing what our general mission is of our research. So we're really excited about the vision or the dream to build artificial molecular machines and motors in the future, which ideally one day will become as sophisticated as those which we have in our bodies. So that's the dream. And of course, there's the vision to also kind of translate discoveries into some real-world applications. So our primary approach in building artificial molecular devices and machines is and really adapting the same principles that nature uses to build up molecular structures, which is self-assembly by folding of biopolymers. So we've mostly worked in the field of DNA nanotechnology, origami, but lately we also ventured out a little bit into the novel protein design, because I think there is some interesting intersection between these two fields, which will yield new insights. So just to make sure everybody's on the same page, I want to quickly explain how programmable nanofabrication of DNA origami works on a very high level. So basically, this is an idea or like a concept in which you can encode three-dimensional structures and sets of DNA single strands. So you can basically sit in your office and follow some rules which have been worked out by the community over the last 20 years or so, and come up with lists of DNA single strand sequences, which as a system will encode a three-dimensional structure. And this is due to partial complementarity between this entire set of DNA single strands. So I have a little illustration here, which is not meant to be physically accurate, to just convey how three-dimensional structures can form from DNA single strands. So in a modality known as DNA origami, you have two different types of components. You have a very long DNA single strand, which has a synthetic sequence, for example, can be several thousand bases long, and you have let's say a couple of hundreds DNA oligonucleotides, which we call staple strands, and they also have design sequences. And these staple strands, they're partially complementary with multiple elements on these strings, basically to different sites on the scaffold strand. So basically, the system tries to minimize this global free energy, and sort of one minimum. The global minimum should actually be represented by the solution, which all DNA strands kind of find their complementary patches on the scaffold. So then, basically, by maximizing the number of base pairs formed in the system, you can kind of get a target structure. So this depends on many factors, like environmental conditions. Of course, there's design rules which will never work, and so on and so forth. But basically, the beauty of this is if you have worked it out, and you basically found a system in which you can fold such structures, it's a really neat nanofabrication method. So in this case, this example, we just produced a nanoscale rod, which is like 40 nanometer long. It has 10 DNA double helices packed in parallel, but it's fully programmable. We know in advance where each DNA base and DNA base pair will be in the context of this three-dimensional structure. It is fully addressable. I can use it as a scaffold to position other types of materials. And you can basically use this very same principle to build a huge diversity of different shapes. So these are some of the objects that we've built in my laboratory over the couple of years. There's actually a movie. So everything which is like yellow and blue is made 100% from DNA sequences, which we have designed. There is no difference between yellow and blue. It just looks a bit nicer. And then sort of what you see here is data. These aren't computer models. These are structures which we actually solved experimentally using single particle cryo-electron microscopy. Initially, by the way, with some help from Shores, who's also working here in the very same building. And So you can build gigantic structures, like a couple of 100 nanometers, like the sun in the center, on also very small structures. And then to give you a sense of scale, I've included here two magenta-colored objects, which have natural origin. We also solved structures of those. So this is a hepatitis B virus core capsid, 35 nanometer diameter. And this is a membrane protein, 800 kilodalton microweight. So basically the point is, with basically modern day DNA nanofabrication, you can come up with all kinds of interesting structures, and they're really commensurate in size to the dimensions of natural macromolecular assemblies. Okay, so that being said, I want to dive into this question of sort of how large can we get and how complex can we get with this kind of nanofabrication approach. So I've been always wondering how far can we push it. Basically, how many DNA single strands can we still program, and how many hundreds of them, and still get like basically a pre-programmed structure. I don't want to bore too much with all the different failed attempts, or like not so optimal attempts that we have, but I want to share one system that we have that we came up with, which we call the programmable icosahedral shell canvas, which is one particular design approach to build up shell-like objects. So it really it's inspired by symmetry properties of natural virus capsids. So we basically adapted this a little bit, and then we came up with sort of what we call a triangular building block. Basically, this is encoded in DNA single strands and self-assembles from DNA single strands. And it yields then a triangular building block, which in this case, this object here is an equilateral triangle, and it has beveled edges. Actually, these are movies, so let me just run. So you can see this. So the edges of these triangles are beveled, and they're also rigid. And they're beveled at a particular angle, so then when you take copies of these triangles, they can kind of stick together like Lego bricks and form a shell, whose geometry is encoded in the geometry of the triangular subunit. And yeah, and speaking of which, basically again, all of this is data. So these are structures which we solved experimentally using single particle tri-EM. So this is one sort of should give you a hint that this actually works quite well, because if you couldn't solve a structure if it's a lot of trash. And then there's one little aspect here, so maybe it's hard to see in the back, but sort of there's these protrusions in recesses. So this is like one thing which you also borrowed from the protein world. It's kind of lock and key or shape complementary docking interactions. So these are basically features which are rigid, and you can use them to control and invent and come up with interaction rules. Basically, how and in which direction should a triangle edge interact with another one. So then you can break the rules. For example, you can change the bevel angle. Then this triangle, which is still equilateral, will make an icosahedral shell instead of an octahedron. You can come up with a different edge interaction rule and also make this an isosceles triangle. Then they will make an even larger shell. In this case, you have 60 copies of the same triangle. And then you can use two different triangles to create different set of local symmetries to make even larger shells. So, and really we were interested in something that works well. And with this system, we had something that worked well. So we can, make these triangles for the icosahedra, for example, and then you get icosahedra mostly. And you can start thinking about what can I do with this. So one thing we did is we used this as a scaffold to position proteins on the outside. So this is just a prototype where we had 60 copies of an IgG antibody positioned on the on the outside of such an icosahedral shell. And again, that's a cryo-EM structure where basically you can see all these IgG with the Fc fragments pointing away from the shell. Why is that interesting? Well, for two reasons, I think. You could think of this, if this would become a future drug delivery vehicle, you could, it's a prototype for showing how to mount, for example, targeting moieties, but it could also become a scaffold for making programmable vaccines, for example. Because you could, in the same fashion like you position antibodies here, you could add antigens from viruses, maybe more, which you make recombinantly, and just add them to these particles. They look like viruses, so they should be able to elicit an immune response. So these are opportunities that you have with this system. There's another interesting thing that you can make with this platform is if you make these shells, and then you can actually modify them and, for example, position two epitopes or two antigens for an IgG antibody in the right spacing across these triangular edges, right? And then sort of add an antibody, and sort of the spacing matches. Basically, that's actually how we fix these antibodies here. Basically, the antibodies, the two paratopes will basically bridge the triangular edge interaction. So you can basically assemble these shells, you can add the antibodies, and you can take these shells away from the conditions in which they were assembled. For example, move them to blood, physiological conditions, but then you can create a situation where they're kind of spring-loaded. So the only thing that sort of holds them together are actually the antibodies. So then if you add soluble antigens, the antibodies will displace, and you can basically trigger the disassembly of the cages. And if you had a payload, you could basically release it on site, triggered by some target protein. And this is a demonstration of this idea. So just for demonstration purposes, we use anti-dick oxygen IgGs to stabilize these shells. So we have a cargo here, which are hepatitis B virus core capsids, just encapsulated in these shells. And then if you add dick oxygenin, which is the antigen for anti-dick, then these shells fall apart and the payload is set free. So you can imagine this might also work in the context in the vicinity of a cell, for example. Then still playing with this, with these systems, since they work well. Another modality is you could come up with this, or you could basically make a modification on these triangle building blocks, which makes them sort of hydrophobic on one specific interface. And then you could imagine a situation where then these triangles bind to a membrane that diffuse around. They'd like to build up the shell, so and through this interaction with the membrane, you might be able to bud off actually a lipid vesicle. Basically, then you would have like a vehicle which is basically single molecule proof or small molecule proof because it sort of has the membrane sealed inside. So here's some some experiments to test this idea. So here's some sort of medium-sized vesicles. You can kind of see here is the two leaflets of the membrane and the cranium, and then sort of here, sort of starting to budding. You can sort of see that budding is in process, so multiple copies of triangles deforming these vesicles, and this is a bit further advanced here. You kind of really bud out. There's a neck formation even here, and then eventually you get to these end products where you have these icosahedra, and then there's sort of a vesicle inside. You can see that, there's a bit bilayer here. And then even there's some instances where we have a scar, there's a missing triangle, which really suggests the way they form is through budding, kind of, where then you get some spontaneous neck scission. So what could be the utility of this? Well, say you bud this off from a vesicle which has a small molecule, which you want to deliver somewhere. You could have that vesicle, and you have a DNA exoskeleton, which is fully addressable. You can add targeting functionalities, so you could even have our release mechanism built in, so you can kind of make more and more complex, future drug delivery vehicles. Then another modality which I want to share is you can kind of you can play with these triangular building blocks and induce shell formation, but do it in such a way that the shells actually don't fully form. You get partial shell products. Like, for example, you could take this triangular building block that makes an octahedron and just deactivate interactions on one of these triangle sides. And then what will happen is you get half octahedra only. And you can do the same thing with the half icosahedron or like a 3/4 of an icosahedron. And again, these are cranium structures, so this works well enough that you can solve structures of these objects. So now you have these half shells. What could you do with the half shells? So we had a wild idea, and this was already 2018 actually, so it's pre-pandemic, but the idea was to sort of, well, these are shells which can actually be larger than actual viruses. What if we take viruses and engulf them? Then just very simply speaking, they should be mechanically unable to interact with cells. And then you basically put them in quarantine. And maybe that's a way to build a universal cell entry inhibitor. So I want to show, basically share a case study where we built basically from the bottom up a chikungunya cell entry inhibitor. So chikungunya is not so, who knows chikungunya? Yeah, so as we expected, it's a mosquito-transmitted virus infection. It's prevalent in the sort of more in the southern hemisphere of the world. It's a very painful disease. You can get very terrible joint pain, and people really hate it when they got it. And there's no treatment for chikungunya, and also no vaccine available. So we picked that as just a case study that we can actually quickly build an antiviral. So we took a shell prototype, which is larger than the typical dimensions of a chikungunya virus. We simply picked an off-the-shelf chikungunya binding moiety, in this case an IgG. We crafted multiple copies of these IgGs to these shells, mixed shells with chikungunya viruses, and then we got chikungunya trapped in individual half shells. And again, that's a cranium structure here of the complex formed between the chikungunya various viruses and our origami. So it works well. Just to illustrate this this is our negative stain T micrograph. So you kind of see here all these surge spherical particles are the chikungunya viruses, and they're kind of all nicely trapped in these shell material. Now, does that actually do anything to virus replication? So we first did some in vitro neutralization experiments with human cell cultures. So this is what you see here is a dose response curve. Basically, a certain dose of viruses given in a cell culture, we titrate in our antiviral, putative antiviral. So the one data curve here, which is interesting, is these gray dots. These are the IgGs, which you purchase off the shelf. So they, by themselves, they don't really have a neutralization. They don't have neutralizing powers. But if you use these compounds as a glue inside of our shells, you do get a potent antiviral, which really, like, it doesn't matter how the IgG binds on the chikungunya. When you use the shell, it becomes a neutralizing compound. And then I think this was very satisfying for us. This idea also works in actual animals. So this is data from July, this is not too long ago. We did some experiments with mice where mice were basically challenged with a lethal dose of chikungunya viruses on day 0, and then they were treated daily, or split in different groups and treated daily with different types of samples. So, for example, there were 2 groups. This blue group here, for example, is basically a group of mice which just got vehicle buffer solution. So, and all of them basically died on day 5, which was the expected result. Then the mice which were treated with the non-functionalized virus traps, meaning they weren't capable of actually trapping chikungunya viruses, also all of them died on day 5. But 100% of the mice which were given our virus trap for chikungunya, all of them survived. So this was almost too good to be true. But so that was the result from that study. And we're in the process of looking deeper and looking at dose response and dosing schedules and so on and so forth. But it shows this concept holds up even in animal. How's my time? Because you have 10 minutes, right? Yeah. That's good. So I can maybe share this thing, which I think is an interesting aspect. So here we go. This this explains. So yeah, this is now something which relates to the pandemic with the coronaviruses. So remember that sort of when the alpha variant came around, sort of multiple companies started with neutralizing antibody program and trying to get it on the market so that we have some kind of antiviral against COVID, right? So one company was Regeneron, and they got a compound approved which was called Regenkov. Donald Trump got it when he was positive, and he said it's like magic. But it was basically a neutralizing antibody which was harvested from a patient which basically recovered from COVID. So in Germany, you also have different efforts of that. So there was one company which was working on a different antibody, and it was a very potent neutralizer for the alpha variant. So this is the vitro neutralization dose response curve again with this antibody from Germany. It's called DZIF10C, and it's a very potent antiviral. If you take that antibody and coat it, use it as a coating in our virus trap, you don't really get a huge benefit. It doesn't make it a lot better. It's already great. But now what happened is that these viruses, they mutated, right? Alpha variant became Delta, and now this hit directly the epitope for these antibodies. The biophysical affinity, binding affinity, just took a drastic hit. So now the efficacy, the potency of this antibody dropped by multiple orders of magnitudes. But interestingly, because of these avidity effects in these shells, you have that same antibody. It's still a binder, right? And we still have to maintain the potency of our virus trap, even up to Omicron. So this idea of using these shells, so another benefit is you can make a very resilient, mutation-resilient antiviral potentially, which can be active for longer. Okay, and then this to basically round us off. I want to show you we played on with all kind of viruses. So we trapped adeno-associated viruses, poliovirus, dengue, noro, human papillomavirus, chikungunya, and SARS-CoV-2 are mentioned, rubella, adenoviruses, and even influenza. And all of these viruses on the same, on this left-hand side, basically they have different dimensions. We just picked a different shell type, but they were all coated with the same compound to trap these different viruses. Because it turns out that many viruses which infect us humans, they have a background affinity to certain polysaccharides, which you find on the surface of mammalian cells. In this case, it's heparan sulfate. So what we did is we just crafted a high density of heparan sulfate to the interior of these virus traps, and then we could capture all these viruses. So there's this vision of maybe we might be able to actually actually built something like a broad-spectrum antiviral platform using this technology. Yeah, so this is the idea. The virus trap is a very straightforward physical approach to build a cell entry inhibitor. So we call this a plug-and-play antiviral, and it remains to be seen whether this actually sort of plays out. But we're certainly going to push it as far as we can. Okay, then I think I have something that I could share with you, but I think I'm just going to stop here and so leave some time for questions. I want to thank a lot of funders who've sort of supported our work for these many years. So I'm very grateful. And then, of course, I want to thank my team for being awesome and making all of these wonderful things with me. And I want to thank you for your attention.
Hi, that's got some really exciting potential on the antiviral front. I was wondering, can you make filaments out of the origami?
You mean dealing with whether we can make filaments from origami? Yeah. You can make tubes and all kind of things. I'm just trying to process where you're heading. Like you're thinking about dealing with filamentous viruses?
Filamentous flu.
Yeah, so we have we have a prototype actually for working with Ebola right now. I'm trying to make something which sort of can deal with Ebola just for. But filoviruses are nasty because they have like a huge surface and a lot of more variability. So one needs to think a little bit outside of a box to deal with that.
Really interesting talk. The virus trap work is very exciting, and I wondered if it would be possible to add the lid of the trap, and I mean either through diffusion or some kind of directed process. Once the virus is in, get the lid on, and would there be any advantage to that in terms of its functionality?
Well, I mean, the way it actually works is kind of that we probably have a certain stoichiometry of sort of a huge excess of these shells over viruses. So you always end up with something packaged by sort of multiple copies, and then you don't really need that lid, right? So you don't really get you don't really gain that much. And I think in some of these images that I showed, you could even see right, there was some human papillomavirus and like two hats, and then it's like kind of so this already happens naturally if you have enough of these shells over viruses. One more question, right, somewhere. Anyway, please go ahead.
A question from online, whether or not the cages induce, the empty cages induce an immune response.
That's a good question. So we have, in passing, touched this a little bit, but we haven't seen anything so far. So what we do is kind of, first of all, this is all extracellular, and part of the sort of innate immune response of foreign DNA sort of intracellularly, right? So whether that's a significant factor. And then from the extracellular side, there might be a sequence-specific immune response for these toll-like receptors, so which would be sort of CG responsive. So we can avoid CG altogether if necessary, but again, we haven't really seen anything. And what we also need to test is sort of whether there's anti-drug antibodies formed, which we'll do in due time. It just takes a longer experiment to figure this out, so it remains to be seen in short.
Another question?
Yeah, just a quick one. Do you co-inject the virus with the particles, or do you treat the mice after they're infected?
So in the one experiment that we did is the mice actually got 10 minutes before they were challenged with the viruses, they got already an intravenous injection of the virus traps. Like you could think of this sort of passive immunization kind of. But sort of we are looking now at all kind of variations, like how long after exposure to the virus, for example, can you still get a therapeutic effect in these kind of things. So this is all ongoing work. Yeah.
One more.
Thanks for the talk. So about the virus traps as well, if it doesn't work as like injected or a taken drug, did you think of using it as a effectively biological filter, if it would like be able to pull viruses from air, and can they be regenerated technically?
I think it's a great idea. We kind of thought about that as well. We just think like the sort of the material costs. Sort of you might be better off like using it sort of as an actual kind of therapeutics or as like as a sort of pre-exposure prophylaxis. So you could also think about nasal sprays, for example, which passivate surfaces in your body which would be prone to get infected or some of this. So these kind of ideas we're thinking about. It's a great idea. We'll see. So we will we'll try to navigate as many opportunities as possible.
Stephen.
I'll try and get this question in before the alarm goes off. So how stable are they? I think in your mice you were injecting every day. So when you put them in, the cages in, how long do they stay in? And then also when you get virus connected in, then what happens, do you think, to the complex?
These are multiple great questions. So we have a post-processing treatment to actually stabilize these traps so that they will actually endure attacks from nucleases and so on in the blood. And the current formulation, or that formulation that we use, is sort of, it lasts for a couple of hours in circulation. It's also why we dose daily, and we're working on that, sort of how to make. We are not even sure where we need longer. Maybe it's good that it sort of disappears quickly. So we kind of, it's like an iterative process. But there is a way to sort of tune the stabilization. It can even be like, stable for a month if necessary. It's just sort of how you prepare it. So right now it's like a couple of hours. And the other question is sort of, sorry, I forgot it.
The other question is, well, what happens?
Oh, yeah, what happened?
Yeah, what happens after when the complex?
Yeah, so what we think what happens is that sort of once the virus is rendered non-infectious, together with this, it forms this complex, it's just being degraded by phagocytes.